Good morning, friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give your valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a very systematic order. I hope this will be very convenient for the continuous study. My dear friends, we are discussing about hepatitis B virus. Last two classes we have seen the history and the epidemiology, and then we have seen the transmission of this particular virus. Today we shall see what is actually the structure of this virus. One end of the full strength strand is linked to the viral DNA polymerase. That means we are talking about the last one, that is genome double stranded DNA. The DNA is there. Now DNA is surprisingly not double stranded, it is single stranded and it is in a circular shape. But important is it contains a DNA polymerase enzyme. Now, one end of the full strength strand is linked to the viral DNA polymerase where it is present. So, it is end to this particular, one end is linked to this particular polymerase. And the genome is 302023320 nucleotides long. How much long it is? For the full strength, full length strand. Genome means the DNA. We are discussing about the DNA. So, the DNA contains nucleotide about more than 3000. And 1700 to 2800 nucleotides are long for the short length strand. Okay, the negative sense, that is non-coding, is complementary to the viral mRNA. The viral DNA is found in the nucleus soon after the infection of the cell. So viral DNA is found in the nucleus soon after the infection of the cell. That means as soon as the hepatocytes are getting infected by the hepatitis B virus, the viral DNA, the DNA of this particular virus, so it will immediately found in the nucleus of the hepatocytes. Now this DNA will use the machinery of the hepatocyte for further duplication. That's the interesting thing. So partially double stranded DNA is rendered fully double stranded by completion of the positive sense strand and removal of a protein molecule from the negative sense strand and a short sequence of mRNA from the sense strand. Ultimately, that single stranded DNA with the help of the DNA polymerase, after entering into the hepatocyte cell, okay, there it gets converted to double stranded DNA. The total story is that. So when double stranded DNA is formed, then it is able to produce mRNA. Isn't it? It is able to produce the mRNA. That means the DNA of the virus is now get connected to the DNA of the hepatocytes. Now it is a part and parcel of the hepatocyte. So whatever the new hepatocytes which are formed, they contain the viral DNA. This viral DNA now produce the mRNA and it will make arrangement according to its need. Very that means very unfortunate here. So non-coding bases are removed from the ends of the sense strand and the ends are again rejoined. So there are 4 known genes encoded by the genome called as C, X, P and S. The core protein is coded by the gene C. The core protein is coded by a gene called as C, HBCAG, and its start codon is preceded by an upstream in the frame AUG start codon from which the pre-core protein is produced. HBeAg is produced by proteolytic processing of the pre-core protein. In some rare strains of virus known as hepatitis B virus, pre-core mutants, no HBeAg is present. The DNA polymerase is encoded by the gene P. DNA polymerase is encoded by the gene P. The gene S is the gene that code for the surface antigen. The gene S is the gene that code for the surface. These are all the genes which are present in the particular DNA of the virus. Okay, so that means these genes are present in the DNA. Now that DNA is able to produce the mRNA. Now by using the machinery of the hepatocyte cell, they keep on producing all these antigens. Okay, antigen P, antigen S, antigen C, antigen E, etc. according to its need. So the HBsAg gene is the long open reading frame but contains 3 frames, starts, ATG, codons that divide the gene into 3 sections like pre-S1, pre-S2 and S. Because of the multiple start codon, polypeptide of the three different sizes are called as large, pre-S1, pre-S2 and S, and middle and the small are produced. There is a myristoyl group which plays an important role in infection on the amino terminal end of the pre-S. S1 part of the large protein. Now, in addition to that, N terminus of the L protein has the virus attachment and cascade binding sites. Because of that, the N termini of the half of the L protein molecules are positioned outside the membrane and the other half positioned inside the membrane. The function of the protein coded by the gene X is not fully understood, but it is associated with the development of liver cancer. Okay, that means this particular gene is there. Now this gene produces its own proteins, virus gene. When it is produced in excess, that may lead to cancerous changes in the hepatocytes. It stimulates the genes that promote the cell growth and inactivate the growth regulating molecules. Now there are many serotypes are there. The virus is divided into four major serotypes: ADR, ADW, AYR and AYW. This is based on the antigenic epitopes presented on its envelope proteins and into 8 major genotypes, A to H. The genotypes have a distinct geographical distribution and are used in tracing the evolution and transmission of the virus. The difference between the genotypes affects the disease severity, course and the likelihood of the complications and response to treatment and possibly the vaccination. There are two other genotypes, I and J, but they are not universally accepted as of 2015. The diversity of the genotype is not shown equally in the world. For example, A, D and E genotypes have been seen in Africa prevalently, while B and C genotypes are observed in Asia as widespread. Now, talking about the mechanism, how this will going to work, and that we will going to discuss in the next class. Thank you very much.